So in the second part of our lab, we would have inoculated our sample. This will be used in lab 2 of Koch's postulates. In this part of our lab, we have our causal organism has been isolated in pure culture. We are observing our organism and we are looking at morphology of the organism. So we are looking at whether the color of your colonies, you would look at the shape of your colonies based on, we did colony morphology in labs 1 and 2. So you can refer to those labs to help with your colony description. We are going to take a sample or a pure culture colony from this sample that has grown, and we will be re-inoculating it into a healthy tomato. We are doing 2 inoculations. One of them is going to be with the unknown organism. The other we will be using a control. So we'll be inoculating a tomato with just distilled water for comparison in this lab. So again, for our procedure, we will be removing the surface tissue slightly to allow for inoculation within your tomato. So sterilize your forceps. You can also do stab inoculation as well using an inoculating needle. Again, your tomato has been sterilized, surface sterilization, to ensure that anything on the surface of that tomato is not going to affect your results, or it's not going to contaminate. The only introduction of organisms we hope is by this unknown organism being inoculated into that fruit. Just remove a really small part. Sterilize your forceps before you put it away. Using your inoculating loop, you're going to sterilize your loop and pick up 1 colony to inoculate your healthy host. Flame till red hot. Alcohol. Flame off your alcohol. Let it cool. So you'll see we have some very tiny colonies here in this region. Taking our loop, we scoop up just 1 little colony. We inoculate it into that region where your tissue was removed. Again, sterilize our loop before we put it back away for future use. Using a marker, we can just highlight the region where your inoculation was done so that you can look for signs and symptoms of growth in that region. So again, we are also inoculating a control tomato using distilled water. These will be placed into Ziploc bags with a cotton bud with some water on it to provide moisture. And this will be incubated for a week. During this period of time, you're going to come in and look for signs and symptoms as the days go along. And that's it for the second session of your lab for Koch's postulates. The causal organism was isolated in pure culture, it was described, and a fresh fruit was re-inoculated with that organism, as well as a control for observation.